Hello dear students. Till now in this chapter we have already studied about the histology of epithelial tissues, connective tissues and muscle tissues. And we know how important all these tissues are. They are specialized for specific functions and together they make the organization function or successful. But have you ever wondered who guides them? Who coordinates them when to function and when not to function? Yes, you guessed it right. There are specific types of tissues which are coordinating all the functions in the body, and these are called as neural tissues. So, today we will study about the histology of neural tissues. We will be studying about these neural tissues again in nervous system later on. Today we will be only studying the histology, structural part, functional part, details of the functional part we will study in another chapter. So let's begin with the characteristic features of neural tissues. First, origin. All the neural tissues, they originate only from ectoderm. They are ectodermal in origin. Then second property, all the neural tissues are non-regenerative, or you can say they are amitotic. They never perform mitosis, but they have another very unique feature. They have a very long life. You know, one neuron may have a life of up to 100 years. Yes. Then next, they are showing excitability, which means they respond to stimulus, and that stimulus should be threshold stimulus, and that threshold stimulus generates impulses which we call as nerve impulses. Okay, so they work on excitability. Then next, not only they get excited, whatever action potential is generated, that is conducted also. So they show conductivity also. So these are the four properties of the neural tissues. Now let's move to the histology. These neural tissues can be divided into two categories: neurons and neuroglia. Neurons are those types of cells which are actually responsible for generating nerve impulse. They are actually responsible for thinking, coordinating, or controlling the muscles, glands. All the function is controlled by neurons. We will be studying about its detail later on. Then next type of cells which are present in neural tissues are called glial cells or neuroglia. These are supporting cells. They are protective, or they also nourish the neurons, and they also are insulating. So the neural tissues are divided into neurons and neuroglia. Okay. Now let's focus on neurons. Neurons are, as I already mentioned, they are amitotic. They do not show any cell division, but they show many unique features. Let us first study the structure of neuron. You can see these neurons. Neurons can be studied in many ways. We will study about the classification also, but let us first study the typical structure of neuron, which you all have been studying from very lower classes. But let us go into little more detail. So neurons, you can see, that can be divided into three main regions. One is cell body, that is called as cytosol. There is axon, and there is nerve ending, that is axon terminal. Let us first talk about this cyton. So this cyton, you can see the cyton is a star-like structure. It's a branched structure. There are many branches. These branches are called dendrites. These dendrites, they branch because they increase the surface area for getting the impulse, for getting excitability. So these dendrites, they receive the information and this information is passed to this part. The cell body has got a very large central nucleus. There are many cell organelles and there are some certain granular parts which are called as Nissls granules. These Nissl granules, they are basically responsible for producing the proteins. They are actually the rough endoplasmic reticulum. Now this cyton now becomes a long fiber like, which is commonly called as nerve fiber. It is cylindrical long nerve fiber called as axon. These axons may be without a myelin sheath, that is non-myelinated, or they may have a protein sheath or a myelin sheath, which are called myelinated nerve fibers. And these myelin sheaths are actually a specific types of cells which are called as Schwann cells. These Schwann cells are actually only the plasma membrane which are insulating the axon. So what are they doing? They are actually helping in very fast transmission of nerve impulse because they do not have transmitting channels. Okay. So these Schwann cells are actually helping in very fast nerve impulses. They are mainly present in our peripheral nervous system where the information has to pass on very fast. Then at places there is no Schwann cells, that gap is there. That gap is called as node of Ranvier. Then this axon terminates into small secretory endings which are called as nerve endings. These nerve endings have got certain neurotransmitters. These neurotransmitters, they pass on the information from one neuron, that is from the nerve ending of one neuron to the dendrite of the next neuron. So this is the structure of the neuron. Now let us study the different types of neurons. So these neurons we can divide into two categories. We can study them based on their structure and based on their function. So when we study them on the basis of their structure, they can be divided into three types: unipolar, bipolar and multipolar. Unipolar neurons are mainly present in the sensory organs. They just take the impulses from the sensory organs. So most of the sensory organs have got unipolar neurons. So they only pass on the information in one direction. That is only from sensory organs to the central nervous system. Then there are bipolar neurons. These bipolar neurons, they are only seen in the eyes and nose. So they have the information, they have two processes. That is, they take information from one side and pass on to the other side. Most of the ganglia present in the retina are bipolar. Okay. But 90% neurons, are multipolar. They take the information from various sources and pass on to various other neurons. So most of them are multipolar. They have many cytoplasmic processes. Okay. Now if we study on the basis of function, then neurons can be divided into sensory neurons, interneurons and motor neurons. Sensory neurons are also called as afferent neurons. They always move towards the central nervous system, or you can say they move from receptor towards the central nervous system. So they take the information from the sensory organs and pass it on to the central nervous system. Then there are motor neurons. They are opposite. They always move out of the central nervous system. That is, from brain or spinal cord towards the either muscles or towards the glands. So they are also called efferent nerve fibers or efferent neurons. But majority of the neurons are interneurons, which are present in between them. They keep on passing the information from sensory neurons to the motor neurons or from motor neurons to sensory neurons. They are interneurons. Okay. So these are the three types of neurons. So with this, we have completed the neural tissues and also the chapter. So chapter is very important. We will be studying all these tissues again when we study the human physiology. So there you have to recall and recapitulate all these tissues again when you study all those different systems in human body. I hope you have understood this chapter very well. Thank you.